Leigh syndrome is most commonly inherited as an autosomal recessive pattern, which means both copies of the gene in each cell must have a variant to cause the disorder. It can have different inheritance patterns. The parents of an individual with an autosomal recessive condition each carry one copy of the altered gene, but typically do not show signs of the condition. Depending on the mutated genome, Leigh syndrome can be inherited as a maternally inherited mitochondrial trait, as an autosomal recessive trait resulting from mutations in nuclear genes encoding mitochondrial respiratory chain complex subunits, complex assembly proteins, coenzyme Q10, mitochondrial targeted tRNA syntheses, and X-linked genes involved in PDHA1 and complex 1, assembly factor NDUFA1, nuclear encoded. Although most patients with Leigh syndrome have a mutation in nuclear DNA, about 25% have a mutation in mtDNA. Leigh syndrome can be caused by mutations in more than 35 different genes of both nuclear and mitochondria origin, involving all 5 respiratory chain complexes.
A mutation in DNA can lead to a malfunctioning protein contributing to conditions like Leigh syndrome. DNA sequence change. Mutations in nuclear or mitochondrial DNA alter genetic instructions, either inherited or spontaneous. Types of mutation, point mutations, can cause missense, altered amino acid, or nonsense, premature stop codon changes. Insertions and deletions. Causes frame shifts, changing downstream amino acid. Large-scale mutations. Duplications, deletions, or rearrangement disrupt gene function. Transcriptions and translations. Mutations affect mRNA production and result in altered protein. Protein folding. Mutations disrupt folding, leading to non-functional or harmful proteins. Protein function. Mutations can cause loss of function, gain of harmful function, or interfere with normal protein. Mitochondrial impact. In Leigh syndrome, affected proteins impair mitochondrial ATP production. Cellular impact. High energy demand tissues like the brain, muscles, and heart are severely affected, leading to progressive degenerations. Overall, DNA mutations alter the amino acid sequence of proteins, affecting their structure and functions, which can disrupt critical cellular process and lead to diseases like Leigh syndrome.
Leigh syndrome is an inherited genetic disorder and one of a broad range of somatic DNA mutations from both parents, or mitochondrial DNA from the mother, that disrupt the normal metabolic function of the cells. Many different genetic mutations can cause this disease, and the specific variants dictate the corresponding malfunctions of the cell. All Leigh syndrome mutations affect metabolic function and decrease ATP production, but most are expressed as a disruption of oxidative phosphorylation in the mitochondrial membrane. Since the mutated DNA encodes RNA, which then encodes the corresponding amino acid sequence in protein synthesis, a variant nucleotide in both chromosomes will result in abnormal function of the target protein. In the case of Leigh syndrome, the related protein could be an enzyme, transport protein, or various other proteins involved in the synthesis of ATP. Sufferers of Leigh syndrome have significantly reduced lifespans, generally dying in early childhood. The most common cause of death is respiratory failure, which develops from both metabolic and respiratory origins. Respiratory failure is synonymous with acidosis, and Leigh syndrome causes an increase of acidity in the cells. As lactic acid accumulates in the cytosol and pH decreases, there is an increased need for gas exchange in the lungs to expel excess acidity and regain pH homeostasis. This acidic load requires the person to breathe more, a task already potentially difficult for those with the condition due to dystonia of the muscles involved in breathing. Lactic acidosis can develop due to reduced oxygen in the cell available for cellular respiration. Chronic respiratory stress will decrease oxygen levels in the body, which causes cells to undergo fermentation in the cytosol to produce ATP and consequently lactic acid. Other forms of Leigh syndrome may directly cause lactic acid production in cells due to disruptions in the pyruvate hydrogenase complex. These variants of the disease block the oxidation of pyruvate into acetyl coenzyme A, causing a buildup of pyruvate in the mitochondria. This concentration increase blocks the continued transport of pyruvate into the mitochondria, thus increasing the pyruvate concentration in the cytosol, where it is turned into lactic acid by lactate hydrogenase enzymes. This pathway to lactic acidosis also increases the need for gas exchange in the lungs via breathing. Without effectively increasing cellular pH, the cells will experience various malfunctions in nearly all cellular functions before ultimately dying. As Leigh syndrome causes decreased ATP synthesis in the cells, certain tissues that have a higher demand for ATP die from this reduced availability. Brain lesions from this type of cell death in Leigh syndrome are a primary symptom of the disease and cause severe neurological deficiencies and inhibited motor control. Other commonly affected tissues are the heart and skeletal muscles. Reduced respiratory rate occurs from a combination of these affected tissues. Lesions in the brain and spinal cord cause a reduction in motor control and consequent generalized dystonia. Together, reduction in related muscle tone inhibits strong breathing, and neurological degeneration can cause abnormal respiration. In short, the acidic effects of Leigh syndrome at the cellular level and its consequent dysfunctions at the organismal level cause acidic inter- and intracellular conditions that are unable to be reduced, leading to death.
Leigh syndrome is caused by mutations in nuclear or mitochondrial genes, impairing cells' mitochondrial function and energy metabolism. According to the most recent research, almost 100 gene variations cause nuclear and mitochondrial DNA mutations, which are the most common causes of the disease in most patients and are inherited by autosomal recessive inheritance, which is the most common, maternal mitochondrial inheritance, which is less common, and the X-linked inheritance. This is the least common. Other cases are caused by genetic variants that are mostly unknown, and more are being discovered. In the case of autosomal recessive pattern inheritance, two faulty nuclear genes are inherited from both parents carrying any of the following genes listed below. Most of these genes are involved in the oxidation of electron carriers, NADH and FADH2, and any fault in one of them will have a direct effect on the respiratory chain and the OX-phos process, and ultimately production of cell energy, which is ATP. In cases that involve PDH complex deficiency, mutations occur in genes that form the PDH complex, affecting any of the three subunits of the enzyme complex and the activation of the complex by PDH phosphatase, eventually leading to lower PDH activity and limiting pyruvate entering the respiratory chain and severely reducing the TCA cycle activity. In the case of cytochrome oxidase deficiency, the nuclear encoded SURF1 gene located on chromosome 9q34 is the most affected by mutation that disrupt complex 4 or cytochrome oxidase. Complex 4 is one of the final steps in the electron transfer in the oxidative process of ATP production, and these strand-creating mutations in the SURF1 gene result in the loss of SURF1 protein, which reduces the formation of normal C or X complexes and eventually impairs ATP production.
Leigh syndrome is a mutation present in both haploid germ cells before fertilization. Because it is carried genetically in the gametes of both parents, our team determined we would need to genetically modify the germ cells before fertilization. One method stood out to us, the CRISPR-HDR modification complex. Since Leigh syndrome has the potential to be present in the variety of single nucleoid base pairs, we needed a tool that could find the exact gene responsible for the condition and one that would accurately rewrite the mutation with the correct sequence exactly. CRISPR, clustered regularly interspaced short palindromic repeats, is an enzyme RNA pair that finds the exact target gene and makes a double cut in both DNA strands to remove the targeted gene. The enzyme Cas9 was derived from bacteria and functions to make the double cut. The partner RNA strand can be precisely synthesized in the lab to complement the target gene sequence, which guides the Cas9 enzyme to allow for the exact specificity of the cut. Once removed, the mutation must be replaced with a healthy version of the gene. This is completed with a donor strand of DNA that acts as a template for rebuilding the removed section. This donor template of HDR, homology-directed recombination, can encode the newly repaired strand with any potential gene sequence. In this case, we would encode the repaired DNA to replace the mutation that causes Lee syndrome with a healthy alternative. After CRISPR HDR therapy of the germ cells, we would continue with in vitro fertilization and gene sequencing of the resultant embryos to ensure no Lee syndrome mutation was present before implantation in the patient.
Testing both parents for mutations and genes associated with Lee syndrome can inform them of their carrier status and the likelihood of passing on the disorder, helping them make informed reproductive choices. Using donor eggs or sperm from individuals who do not carry the mutations associated with Lee syndrome can prevent the transformation of the disorder. Couples at risk of having a child with Lee syndrome can benefit from genetic counseling to understand the risk of the available reproductive options to avoid passing on the disorder. These approaches can help reduce the risk of passing on Lee syndrome to offspring by ensuring that the germites used for conception do not carry the harmful mutations responsible for the disorder.